Hello and welcome to this video covering the content of the GCSE AQA Biology course. Over the next couple of months on this channel, I will be covering all of the content that you need to know for your science exams, so please subscribe if you don't want to miss out on all that. In this video, we are covering the first topic of the course, cell biology. Timestamps for your science course are in the description. Let's begin. So before we start, it's worth taking a look at the advanced information that AQA have given us for these exams. So they've given us the topics that the questions are going to focus on and the topics that aren't going to be in it at all. So if you're doing the biology, physics and chemistry as separate sciences or triple sciences, I'm going to refer to them throughout the video, and you're on the foundation course, cell structure and cell transport are going to be major focuses, while cell differentiation, which is part of cell structure, won't be included at all, and two practicals, microscopy and osmosis, are going to be assessed. So stuff that isn't a major focus, but it's also not assessed, if that makes any sense. For example, 4.1.2 cell division, that might come up, so you might get asked a few small details about it, but it's not going to be the focus of one of the big questions. So it's still worth paying attention. Also note that in this video, cell biology, that's all the 4.1 stuff that we're covering. So here's the list for triple science biology higher. So similar cell structure and cell transport, and the two required practicals, but cell differentiation will be coming up. For the Combined Science AQA course, it's a different story because in Foundation, cell division is coming up, as is the microscopy practical, but the osmosis isn't coming up at all, and neither is active transport, while the rest of it's a diffusion and cell structure that might come up. Combined science higher, similar again, cell division will come up, but this time microscopy for you guys won't be in it at all, and neither will any cell transport stuff, so you actually have a lot less to learn and you can focus your revision a lot more on cell division, but still pay attention to the cell structure stuff. Okay, we're going to start by looking at cell structure. So the first thing to be aware of is the two types of cells. So we have eukaryotic cells and prokaryotic cells. So eukaryotic cells are basically just cells where the DNA is contained in the nucleus. So here you can see a cell, animal and plant cells here, and you've got the nucleus, the DNA's inside it. They're both eukaryotic cells, whereas prokaryotic cells have a single loop of DNA that floats freely in the cytoplasm. So as you can see here, this is a bacterial cell, this is a prokaryotic cell. It's got this DNA loop as well as these plasmids. It's worth noting that an organism made up of eukaryotic cells is called a eukaryote, whereas similarly a prokaryote is made up of prokaryotic cells, but prokaryotes are single-celled organisms and they are just bacteria, so bacteria single-celled. Another important difference is that eukaryotic cells are bigger, prokaryotic cells are smaller, and inside bacteria cells, obviously you have the DNA loop, the plasmids, which are just small rings of DNA which can replicate themselves. There's also the cytoplasm, this jelly-like substance where the chemical reactions happen. It has a cell wall to protect the cell, a cell membrane to control what enters and exits, and bacterial cells also have ribosomes, which makes protein through protein synthesis, but they don't contain mitochondria or chloroplasts, which is unlike animal and plant cells. So let's look at those. Here on the left we have an animal cell, and you can see it has the cytoplasm, it has a cell membrane, it doesn't have a cell wall, but it does have the ribosomes that produce protein. It has mitochondria, and these provide the cell with energy through a process called aerobic respiration, which comes up later. And keyly, because they're eukaryotic cells, they have a nucleus which contains the genetic material of the cell, and it also controls the cell's activities. Now, plant cells over here on the right, they contain all five of these things, but they also have a cell wall, which is made of cellulose to support and strengthen the cell, and they have a massive thing called a vacuole in the middle of the cell, and this has various functions, but the one that you probably should remember is that it contains the cell's sap. And plant cells also contain chloroplasts, which contain a green pigment known as chlorophyll, and this absorbs the light for photosynthesis, so chloroplasts are where photosynthesis happens. So now we're going to look at microscopy, which is especially important for the triple science people. So there are two terms that are very important when understanding microscopy. Magnification is how many times larger the image is compared to the real object, and resolution is the ability to distinguish between 2 points. So you can think about it as the quality of the image. You may have heard it be used to describe computers. So there's also two types of microscopes. There's light microscopes, which you have in school, which pass light through an object. We call them specimens when we're looking at them through microscopes, and they use lenses to magnify it. So we can see cells and their nuclei through these. But there's more expensive advanced microscopes called electron microscopes, where we pass electrons through the specimen, and this allows us to see really small subcellular structures like ribosomes in detail, and this just helps us better understand how subcellular structures work. Subcellular structures being the things inside the cells, so like ribosomes, mitochondria, nucleus, etc. And it's important to remember that electron microscopes have a higher magnification and resolution. You also need to be able to calculate the magnification of an image. So here's the equation for it. Magnification is the size of the image divided by the size of the actual object, and size, that could be height, width, length, whatever. It'll give it to you in the question. I find a good way to remember this is that the real object times by the magnification gives you the size of the image you're looking at, and we're going to look at an example of this. So, in the example question, we're told that a light microscope with a magnification of times 50 creates an image of a xylem cell that's 2.5 centimeters in height. So this is our image size, this is our magnification, and we are asked what the actual height of the xylem cell being observed is, and we're also asked to give our answer in micrometers. So we're going to start by rearranging the equation, which is something that you should be good at from maths. So remember the equation first. So you have the real size of the object multiplied by the magnification gives you the image size. And then we divide by magnification because we're looking to get the real size. So the real height is equal to the image size divided by the magnification. Substitute our values in now. So our image size is 2.5 centimeters, and that's divided by the magnification of times 50, and that gives us a result of 0.05 centimeters, but we would need our answer in micrometers, so we're going to times by 10 to convert to millimeters, and then we can times by 1,000 to convert to micrometers. And there is our final answer of 500 micrometers. So now we're going to move on to the microscopy required practical, which is coming up in the triple science exams and the Combined Science Foundation exams. So there's 3 stages to this. It's quite lengthy. We have preparing the specimen, we have actually using the microscope, and we have drawing what you can see. So starting off with preparing our specimen, you're going to take a microscope slide and you're going to place a drop of water in the middle of it, and you're going to place the cells that you're studying into that water. And then you're going to add a drop of iodine, and this acts as a stain which allows you to see the cell more clearly. Always remember that you could be asked why you do any stage of the required practical, so remember you understand why we're adding iodine, why we're not trying to trap any air underneath the cover slip for our next stage, which is take the cover slip, because that will make the specimen much harder to see. So stage 2, using the microscope. And it's first important that you know the many different parts of the light microscope. So the bit that you look through is called the eyepiece lens. These 3 lenses down here are called the objective lenses. You've got the stage, which is where you put your sample, as well as your slide clips. You've got the light, which is attached to the base, and you've got the arm here. And then the 2 bits that are important that you might not know about. This is the coarse focus, this knob here, and this small one is the fine focus, and they're important in this practical. So the first stage is quite obviously to use the slide clips to hold our sample in place, and then we're going to select the objective lens with the lowest power so that we can identify and zoom in on the bit that we want in the future. So we're going to use the coarse focus first to move the stage up until our specimen is roughly in focus, Then we're going to use the fine focus to make sure it's completely in focus. And once it's completely in focus and we can see it, we can switch to an objective lens with a higher magnification to zoom in on any part of the cells. And then we need to draw it. So essentially, you just need to remember how to keep it accurate by using a sharp pencil, drawing unbroken lines, filling up at least half the space on your page, draw labels, and also you can draw a scale bar. And this has come up before, so I'm going to show you how to do it. So the first step in creating your scale bar drawing is to find out the actual size of a single cell that you're observing. So to do that, you're going to take a clear plastic ruler with millimeters on it, and you're going to put it under the microscope next to your sample, so that hopefully when you look into the microscope you can see how many cells fit into the gaps between the lines. So you get 4 cells in a millimeter. So if you've got 4 cells in 1 millimeter, which is 1,000 micrometers, and if you divide both sides by 4, think of it a bit like a ratio, then you're going to have 1 cell is 250 micrometers long. So once you know the actual size of your cell, you want to know the size of the image that you've drawn. So take a ruler, measure one of the cells that you've drawn. This one is 8 centimeters. So you know that on paper, your 8 centimeter drawing is equivalent to 250 micrometers. So if we want to make like a key, we'd say that 1 centimeter is, and you treat it like a ratio again, so 8 divided by 8 is 1, 250 divided by 8 is 31.25. So you can draw a little key there stating that 1 centimeter on your paper represents 31.25 micrometers. Okay, on to a quick bit about cell differentiation. This is pretty easy to understand. It's mainly just a definition, really. So cell differentiation is where a cell changes and develops new subcellular structures to allow it to perform a specific function. It's essentially when your average cell turns into, I don't know, a nerve cell, or the specific cells that you hear about. And undifferentiated cells are called stem cells. We'll look at them later. And you need to remember that in animals, all our cells differentiate early. So we find a lot of stem cells in the embryo, but as adults, we don't have many undifferentiated cells. We have some, but they can only differentiate into specific cells. Plants, however, they can differentiate throughout their lifetime as they grow, and their stem cells are found in the tips of stems called the meristems. Again, we'll look at this later. And cell differentiation, it's used to grow new tissue or repair damaged tissue in us, in humans. And then we have a few case studies, if you like, of cells. We've got to look at sperm cells, nerve cells, muscle cells, root hair cells, xylem cells, and phloem cells. Sperm cells, first of all, obviously they have this tail, it's called a flagellum, and it allows the cell to move itself towards the egg. They have lots and lots of mitochondria to provide enough energy for this journey to the egg cell. They only contain half the usual amount of chromosomes, so that when they fuse with the egg cell, they can share DNA. And finally, this thing here is called an acrosome, and it contains enzymes which allows it to fuse with the egg. With nerve cells, it's all about transferring electrical signals around the body, so that they're long, so they can cover more distance. And they also have this bobbly protective myelin sheath, and this keeps the electric signals inside the cell while they're passing through it. And then all these branches around either end, they connect to other nerve cells to form a chain to pass the electric signal along. The role of a muscle cell is to contract and produce movement. So first of all, they're long so that they have the space to contract, and they contain protein fibers which allow them to contract. And the mitochondria generate energy for the contraction. So here we have a root hair cell on a plant, and these are basically, they're on the root, and their job is to absorb water and mineral ions. They have a large surface area, as we'll look at later. This means that they can absorb a greater volume of water and mineral ions, and they also have no chloroplasts because they're underground. Moving on to our final 2 cells, xylem and phloem cells form xylem and phloem tubes which go through plant stems and transport water and minerals. Xylem transports water and phloem transports minerals, so remember xylem and phloem, water and minerals. They both have open ends to allow substances to flow through them, and they also have few subcellular structures which also allows substances to flow through them. And xylem cells contain lignin which strengthens the walls. So it's time for a quick memory test to see what you can remember from the previous slides. Pause the video, think of the answers in your head or write them down, I don't really mind. So in bacteria, there's obviously the DNA loop and plasmids, they're the most important, but did you also remember the cytoplasm, the cell wall, cell membrane, and the ribosomes? The genetic material is floating freely in the nucleus as loops of DNA and plasmids. There's three differences. Eukaryotes are bigger. Eukaryotes have the genetic material contained in the nucleus, and eukaryotes are multicellular, whereas prokaryotes are single-celled organisms. Plasmids are small loops of DNA in bacteria cells that can replicate themselves. Resolution is the ability to distinguish between 2 points. So this next bit on culturing microorganisms has never been in the Combined Science course, so it's only for triple science people, and we're going to have a closer look at bacteria. Now, bacteria multiply using something called binary fission. It's quite similar to mitosis. So the first stage is the genetic material in the bacteria replicates. So that's obviously the DNA loop and the plasmids, and they move to opposite ends of the cell, just like in mitosis, and the cytoplasm divides, a new cell wall forms over it, and the two new cells that form each have one circular strand of DNA, but a variable number of plasmids. Now, another thing to remember is that bacteria need the right conditions to multiply. They need a warm temperature, preferably 37 degrees C, that's optimum. They need moist conditions, they need food, they need time, they need the right pH, which can vary between bacteria, and in optimum conditions they multiply at a rate of once every 20 minutes. In the lab, we can grow bacteria in two ways, in an agar gel plate or in a nutrient broth solution. You will have probably done it in an agar gel plate at school. So there's a required practical which isn't coming up in anyone's exam, where you add paper discs soaked in antibiotics to an agar gel plate growing bacteria, and you see how large an area around the disc becomes bacteria-free, and this zone is known as an inhibition zone. And the inhibition zone is, think of it as the area in which bacteria is inhibited from growing. You could be asked on that, and you can calculate the area of the inhibition zone by using pi r squared. Now, a more complex calculation that I'm going to cover here, even though it might not be important, is the calculation for the number of bacteria that have multiplied in a specific time period. So a question might ask you, how many bacteria are there after an hour, so after 60 minutes? So first you have to work out the number of divisions that happen in the time period. So our example that we're doing is 60 minutes. So let's say, for example, that bacteria are multiplying at their optimum speed of 20 minutes. So there's 3 20s in 60, obviously you have to divide one by the other, so 3 divisions happen in an hour. So you do 2 to the power of this number, as each division through binary fission produces 2 bacteria. So after the first division there's 2, second times by 2 there's 4, third times by 2 there's 8, 16, 32, etc. So 2 to the power of 3 is 8. And then if you're told that you have a certain number of bacteria already in the colony, for example, I don't know, there's 350 bacteria to start with, you would multiply 350 by 8 to get your final answer of 2,800. You also need to know aseptic techniques that we can use to prevent contamination. So contamination, we just mean that bacteria that isn't part of our investigation starts to grow in our investigation, and this could be potentially harmful to us, so we want to prevent it using aseptic techniques. And aseptic simply means free from contamination. So here's not a hard definition to remember. We have 3 important aseptic techniques. Keep the maximum temperature to a maximum of 25 degrees Celsius. This is 12 degrees below the optimum of 37, if you remember that from the previous slide. All equipment should be sterilized before use, so we can usually done by heating the equipment to a temperature deadly for bacteria. This includes the Petri dish, the culture medium, and the inoculating loop, which is a loop that we use to carry bacteria. Go look it up if you don't know what it looks like. We can pass it over a flame to sterilize it. And finally, the Petri dish should be placed upside down and taped on to prevent condensation falling into the experiment with potentially harmful bacteria inside. So now we've made it to the end of cell structure. Here are some questions to test your memory. Try and only answer the ones that are coming up in your course. So pause the video, say them in your head, write them down, whatever. So for 1, you have the permanent vacuole, which stores the cell sap. You have the chloroplasts, which contain chlorophyll, which absorb light for photosynthesis. and you have the cell wall, which is made of cellulose to protect the cell. Respiration happens in the mitochondria. Magnification is how many times bigger the image is compared to the actual object. Electron microscopes have helped us better understand subcellular structures by allowing us to see them functioning more clearly. The equation for magnification is the image size divided by the real object size. The 3 stages are the preparing of the slide, the using the microscope, and the drawing what you can see. The steps to the second stage is you clip your slide on, you use the objective lens with the lowest magnification, you adjust the stage using the coarse focus and then the fine focus to get the cells into focus for you to see, and then you can use a higher powered lens. Cell differentiation is where a cell changes and grows new subcellular structures to allow it to perform a specific function, and it happens in plants throughout their lifetime. Nerve cells have that protective myelin sheath to keep the electric signals inside the cell. They're long to cover more distance, and they have branches called dendrites to connect to other nerve cells and pass the signal on. Root hair cells have a large surface area, so they can absorb a greater volume of nutrients and water, and they also don't have chloroplasts, in which the two mediums are an agar gel plate or a nutrient broth solution, and aseptic is simply free from contamination, and the free aseptic techniques: sterilization, maximum temperature of 25 degrees C, and keep the Petri dish lid taped on and upside down. So now we're at the cell division bit. So if you're doing combined science, this is very important. This is going to make up about 1/3 of your paper, so pay attention to this next slide. So inside our nucleus we have 23 pairs of chromosomes. That's very important. Memorize that fact for both biology papers and even the chemistry papers. And chromosomes contain our DNA. So we're going to look at mitosis first, and then we're going to go on to stem cells. So mitosis, or should I say the cell cycle, it happens in 3 stages. So the first stage is preparation, where the cell increases its number of subcellular structures and duplicates its chromosomes. This just means there's 2 copies of each, and this stage takes longest. They might ask you about that, I don't know. And then stage 2 is the mitosis stage, where the chromosomes, they go to opposite ends of the cell, and the membrane forms around them so that there's now two nuclei in the cell. We say that the nucleus divides. That's enough for GCSE level. And then the final stage is the actual division part, where the cytoplasm and the cell membrane split and divide to form 2 new cells. Mitosis, it's important to remember, it produces 2 genetically identical daughter cells, which can be used for growth or to repair damaged cells, and that's important for both paper 1 and paper 2. So now stem cells, again we briefly touched on them when we went over cell differentiation. Stem cells are just undifferentiated cells. So in adults, stem cells are only found in certain places. They can only differentiate into a limited number of cells, and we already use them in medicine. So for example, they can differentiate into blood cells or skin cells to heal your grazes or something. Embryonic stem cells get scientists excited because they can differentiate into any type of cell, which means we can in theory use them to cure things like diabetes and paralysis by growing new cells. We grow them through therapeutic cloning, which allows us to grow an embryo with the same genes as the patient, so that the patient's body won't reject the new cells, like in heart transplants, for example. However, embryonic stem cells have ethical objections, as people believe every embryo is a potential human life, and destroying that human life to use it to heal someone is wrong. There is also a risk that stem cells can become contaminated by a virus when we grow them in a lab, and that could put a patient at risk of being ill even more. In plants, obviously you know that plants can differentiate throughout their lifetimes, and they have stem cells in the meristem tissue, and this is like at the tip of the stem and the parts of the plant where growth happens. We use them to clone plants quickly and cheaply. We can use them to grow rare species to prevent them from extinction, and we can also clone plants with desirable traits such as disease resistance. So 3 uses there to remember. So that was all of the content in the cell division topic, so there's not really much to remember there. Here's a quick Can you remember test? See what you can remember from the previous slide. Pause the video, have a go. So, function of the vacuole, it has various functions, but the most important one is that it stores the cell's sap. Ribosomes make protein through protein synthesis. Our body has 23 pairs of chromosomes. The 3 stages of the cell cycle are preparation, mitosis, and division. In preparation, the cell increases its number of subcellular structures and duplicates its chromosomes. In mitosis, the chromosomes go to opposite ends of the cell and the nucleus divides. And in division, the cell membrane, not the cell wall, and the cytoplasm divide. 3 sources of stem cells, we can find them in adults, in the bone marrow, for example. We can find them in embryos, and we can find them in the meristems of plants. Remember, those are the parts of the plant where growth happens. The answer to number 6 is because adult stem cells can only differentiate into specific cells, whereas embryonic stem cells can differentiate into any kind of cell. A disadvantage of embryonic stem cells is when we're growing them, they can become contaminated with a virus and make a patient even more ill. And the final question, an important 3 uses to remember: we can use stem cells to clone plants quickly and cheaply. We can use them to clone rare plants, which prevents them from going extinct. And we can clone plants with desirable traits like disease resistance. So, now we're going to move on to cell transport. This is a big topic in the triple science paper, but in the Combined Science higher it isn't coming up at all, so you want to skip it. Combined Science Foundation you want to skip after diffusion. So diffusion, it's just where particles spread out from an area of high concentration of those particles to an area where they're lower concentrated. So imagine you spraying some perfume and it spreads out across the room. That's just diffusion. It's also how substances travel into and out of cells. And diffusion works with the concentration gradient, and that is simply the difference in concentration between 2 areas. Factors that affect the rate of diffusion: the bigger concentration gradient, the faster the rate of diffusion; the higher the temperature, the faster the rate of diffusion, as the particles have more energy; and the larger the surface area, the faster the rate of diffusion. And that's like if diffusion is happening through a membrane, as this means that more particles can be transferred at once through the larger surface area. We're going to look at 2 functions of diffusion. Our body uses it in gas exchange, so carbon dioxide diffuses out of the lungs, oxygen diffuses into the lungs through the alveoli, which we'll look at later. And urea, that's a waste product that comes out through the kidneys, that diffuses out of cells into the blood, and it's taken to the kidneys for excretion. So surface area to volume ratios, they're quite easy to work out and compare. You just calculate the surface area and calculate the volume. You should know how to do that from maths. And then you can convert the ratios so that the volume ratio is the same on both, and the one with the larger surface area is referenced to as having a larger surface area to volume ratio. So now we're going to look at the need for exchange surfaces. So while single-celled organisms like bacteria rely on diffusion to get substances into and out of the cells, we, because we're multicellular, can't do this because lots of our cells aren't exposed to the air, so we can't just absorb oxygen because our cells don't have access to any. So we need exchange surfaces and systems of transport. This is why our cell membranes are partially permeable. It keeps the contents and shape of the cell, but it also has small holes which allow molecules to diffuse across it. So let's look at the conditions that make exchange surfaces more effective, because in a minute we have another 5 exchange surfaces that we've got to look at. So a larger surface area to volume ratio allows more of a substance to diffuse into a space of the same size. A thin membrane, usually 1 cell thick in most of these cases, means that substances have a shorter distance to diffuse over. Lots of blood cells allow substances to be absorbed into the blood, and this is only where substances in the blood need to be exchanged. And where gases are being exchanged, good ventilation maintains the high concentration gradient. So now let's look at our exchange surfaces. Sorry, I can't be bothered to draw pictures for all of these. Some of them are just too complicated. I'm not drawing a gill. So alveoli, they have an enormous surface area, and they're only 1 cell thick. By the way, these are in the lungs to allow the exchange of carbon dioxide and oxygen. And they also have a very good blood supply. Villi are their small projections in the small intestine there to absorb digested food. Again, they have a large surface area, one cell thick walls, and a very good blood supply on the inside. Leaves have two exchange surfaces on them. As you may remember, the bottom of the leaf is covered in stomata, which are used for exchanging carbon dioxide and oxygen between the leaf and the air, because the carbon dioxide is needed for photosynthesis. So the flattened shape of the leaf, that provides a large surface area for this exchange to happen, while in the inside of the leaf, carbon dioxide needs to get into cells, and the air gaps inside the leaf provide a large surface area so that more of the carbon dioxide can be absorbed into the cells in the leaf. To absorb water and minerals, roots have root hair cells, as we talked about earlier, which provide a very large surface area to volume ratio, and they also have thin membranes. Again, if you get asked a question about one of these, referring to this list of four, you'll always get marks from it. So finally, the gills in fish, probably the hardest one. So in the gills, the aim is to absorb oxygen from water and get rid of carbon dioxide. So what they have in the gills is these lots and lots of little plates called gill filaments. These give the gills a massive surface area, and because these gill filaments have a great blood supply, the blood removes the oxygen and supplies carbon dioxide, while the water takes away the carbon dioxide and supplies more oxygen, and this maintains the high concentration gradient, so you could call it good ventilation. So now we're going to move on to osmosis, and osmosis is diffusion, but it's only with water over a partially permeable membrane. So be careful with the definition. It's the net movement of water molecules from an area of higher water concentration to an area of lower water concentration through a partially permeable membrane. Always check whether the question is talking about the water concentration or the concentration of molecules in the water. If we take sugar, the water is going to be moving from areas where there's a low sugar concentration to a high sugar concentration, because where there's a high sugar concentration, there's going to be less water. And this is important for the osmosis practical. Also, a note about the net movement. That just means that the water molecules are actually moving both ways through the membrane. It's just that the side with more water on will have more water molecules moving out of it. The osmosis required practical, and again, this is the second required practical of the video for the triple science people. So you take an apple corer or something similar and basically just use it to cut a potato into 5 identical cylinders. And this is important because we need the surface areas to be equal, so this is a control variable. Measure and record the mass of each using a balance. Fill 6 boiling tubes each with different sugar solutions, different concentrations of sugar solutions. So 1.0 moles per decimeter cubes, that's very concentrated, and you can go down by a certain margin each time. And 0 moles per decimeter cubed is pure water. So then you're going to add a potato cylinder to each one of the boiling tubes. So if you're using 6, you're actually going to be needing 6 identical cylinders. My bad. And leave them for a day. That's important because osmosis doesn't happen instantly. It takes a while. So you then, when it's finished, you need to dry each cylinder gently and just roll it over a paper towel or something. Be really gentle with it, but also make sure that you get the water off. That could be a question when they ask you about accuracy of this. Have you got all the water off? Is that affecting the end result of the mass when you measure it? And now the final thing to do is, because the identical cylinders might have slightly different masses, calculating the percentage change in mass, and then you can plot a graph with concentration on the x-axis, percentage mass change on the y-axis, and you can use this to find the sugar concentration of the potato roughly by drawing a line best fit and reading off the concentration where the percentage change would have been 0. So our final method of transport between cells is active transport. This is again different from diffusion because it's the movement of molecules from an area of low concentration to an area of high concentration this time, so against the concentration gradient, and it requires energy, so diffusion but backwards. It also requires protein carrier molecules. These transport the molecules. So 2 uses of active transport. In plants, root hair cells use active transport to absorb mineral ions because there's a higher concentration of minerals inside the plant than outside. And we use active transport in the gut to move digested materials like glucose and amino acids from the gut into the blood, as there's a higher concentration of these things in the blood than there is in the gut. Generally, we get the energy for this from respiration. So finally, we're going to briefly look at what the differences between diffusion, osmosis, and active transport is. So active transport is different because it goes against the concentration gradient and it requires energy, and osmosis is different because it requires a partially permeable membrane and it only moves water, unlike diffusion, which can just happen through air, like you can see here with this diffuser. These particles will just diffuse out through the air. So, now we've covered all of the methods of transport in cells, pause the video and have a go at these. Diffusion is where particles spread out from an area of high concentration to an area of low concentration. You increase the rate of diffusion by increasing the surface area of the membrane if it's diffusion across the membrane, increasing the concentration gradient, or increasing the temperature so that the particles move faster. Multicellular organisms need exchange surfaces because not all of our cells are facing the outside, and we can't absorb things like oxygen because our cells don't have access to that. How can exchange surfaces be made more effective? A thin membrane, a good blood supply, good ventilation, and a larger surface area to volume ratio. Those are the four things. How are leaves adapted? Well, remember there's two exchange surfaces on leaves. The outside of leaves, by the stomata, they have a flattened shape to have a higher surface area, and inside leaves there are gaps to increase the surface area of the leaf cells to allow more carbon dioxide to be absorbed for photosynthesis. Gills in fish have gill filaments, which have a high surface area. They're thin, they have a good blood supply, and remember all that water coming in, that removes the carbon dioxide to keep the concentration gradient high, so good ventilation as well. Osmosis is where water moves from an area of high water concentration to an area of low water concentration through a partially permeable membrane. The dependent variable is the one that we measure, and that is percentage change in mass in this case. A control variable is the surface area of the potato cylinders. Active transport requires energy and protein carrier molecules. Active transport is used to absorb minerals in root hair cells and digested substances in the gut in humans. And a similarity of diffusion and osmosis, that would be that they both move with the concentration gradient from an area of high concentration to an area of low concentration. So now that we've made it to the very end of the video, we've covered everything in cell biology, and depending on what your exam will feature, you will know exactly what's coming up. So now, how do you revise it? Well, these 3 slides of questions contain every single bit of knowledge from the PowerPoint. So what I suggest is that you do the questions from these 3 slides that are relevant to you. So in the case of triple science people, that's going to be cell structure and cell transport, but also give these a go because they might come up. For combined science higher, you will want to have a go at maybe the first 4 of these, but definitely skip the magnification and microscopy questions. Cell differentiation questions are important, and remember, only have a go at the culturing microorganism questions, question 6 onwards, if you're doing triple science. Cell division, this is so important to anyone doing combined science. If you don't do anything else, revise this. And cell transport, again, this is only coming up in the triple science exam. If you're doing combined science foundation, it would be worth covering the diffusion and exchange surfaces stuff, but if you're doing combined science higher, ignore all of these completely. So what I suggest is, once you've done them, any that you get wrong, note down and come back tomorrow and have another go. You're going to go through the video to check, by the way. And then a week from now, come back, have a go at them, and then once again, come back to the ones that you get wrong the day after. And then along with some exam practice questions, maybe nearer the time of the exam, you should be able to nail all of this knowledge and get a fantastic grade in the summer. So these are the cell structure questions. There's 28 if it had let me number them properly. There's 11 cell division questions. Lucky combined science people, you don't actually have to revise that much. and the cell transport questions for the people doing triple science. There's another 22 of these. If you don't want to miss out on future ones of these, because we've got another 3 biology ones coming, we've got 5 chemistry ones, and that's just for paper 1. So there's a lot of knowledge that you need to know. So subscribe if you don't want to miss out on those. Thank you for watching, and I hope this has been helpful.